There we go. So I'm going to do a quick review, like I said, with my whiteboard now, since I'm back in action. So I'm going to just draw a basic brain structure. If this is anterior, the front, this is posterior, the back. So the person actually is facing this way, our left as we face this screen. So I'm just going to draw a basic outline of the cerebral hemisphere and identify just the major landmarks. Remember that a gyrus is one of these raised ridges that we see all over the brain surface, and then a sulcus is just one of those grooves created between the gyri or two gyrus. A fissure, remember, is just a deeper groove than a sulcus. The sulcus are shallower grooves, the sulci. All of that stuff, especially the gyri and the sulci, excuse me, it's the first time I've spoken today to anybody. All of those things increase brain surface area. That's their main function, which obviously increases functionality. So just to go over the major landmarks that we mentioned yesterday, that median longitudinal fissure can't really be seen. The median longitudinal fissure can't be seen from this lateral view that I have started to do. But if we were to look at somebody's face from the front, the brain would look something like this. And there's that long fissure separating the right half from the left half. That's the median longitudinal fissure. The central sulcus, which separates the frontal lobes from the parietal lobes, goes like this. It's from this lateral view that I started to draw in the big picture. There's a central sulcus right about here. It's not as deep as a fissure, but it runs down this line and it helps us define this primary motor cortex. I'm just going to put PM for primary motor cortex from this, and I'm using the same colors as your book, from the primary somatosensory area. So that central sulcus separates the motor from the somatosensory areas. That helps quite a bit. The lateral sulcus, which is what I was trying to explain to you yesterday on my own head, that runs up something like this. So just to review real quickly, the median longitudinal fissure is this one that separates right and left halves of the brain. The central sulcus is this one that runs down, and it goes down both sides of the head. So it would sort of roll over our head like a headband, like a girl's headband that's worn way up high, pushing her hair back. And then the lateral sulcus here, that sort of separates the temporal lobe down here from the parietal lobes up here. So that just goes through the major sulci, fissures, the major landmarks of the brain. It helps us find things. Now I'm going to clean up my drawing really quickly and show you how those ventricles are positioned in the brain. I'm going to use blue again just because that's the color that your book uses. Remember, the ventricles, the brain ventricles, are just spaces or cavities that house cerebrospinal fluid. And that's initially where the fluid is secreted. So we've got two big, I'm exaggerating this. That's actually, that's kind of ridiculous. Let me erase that and start over. They're not nearly that big. They're about this big in the center of the brain relative to the rest of the brain size. So we have these two big alien looking things that are in pair, in tandem, that one on the right and one on the left. So if I draw it from the side view, you just barely see the other one on the other side behind it like this. We call those the lateral ventricles, and they're ventricles 1 and 2 of the 4 major ventricles. So this one and this one on the right and left side would be the lateral ventricles. I'm just going to put in parentheses 1 and 2. And their spaces, this is all a continuous space. They're filled with fluid. Has this little hole in it right here. Down below the two lateral ventricles, or ventricles 1 and 2, is the third ventricle in this area that I'm coloring in dark blue. So this is the third. And then there's something called the cerebral aqueduct, this thin hollow tube filled with cerebrospinal fluid that leads to the fourth ventricle, which kind of looks like a tail of this structure. So that's how these ventricles sit in the middle of the brain, and they're spaces that are filled with that fluid. And this is how I was trying to describe to you yesterday, and it wasn't ideal because I didn't have this board. Associated with each of these four ventricles is a small knot of capillaries, very tiny blood vessels. So there's one underneath here, as well as the other one on the other side just next to it. There's a knot of capillaries associated with the third ventricle and another one with the fourth. Those capillary networks or knots are called a choroid plexus, and it's just a knot of capillaries that pushes, if they're under high pressure. And being under high pressure, if this is, I'm going to magnify this, if this is a blood vessel or a capillary that's under high pressure, fluid from that capillary gets pushed out of the wall of the capillary like this into this space of a ventricle. And that's where cerebrospinal fluid comes from. Then that cerebrospinal fluid, it circulates throughout all four ventricles, and it circulates down through the center of the spinal cord, down through the brain stem and the spinal cord, and then it makes its way up. I'm going to draw the spinal cord here. So it makes its way down through the center of that, and then it exits the spinal cord and sort of bathes around the outer surface of it and tends to circulate back up, and it also circulates this fluid underneath the arachnoid matter, which is the second layer of protective coverings around the brain. So this fluid goes all over the place, is what I was trying to describe yesterday, but its derivation really is here at these choroid plexus knots and the ventricles. Okay, so that hopefully gives you a little bit better idea of what's going on with the ventricles and the CSF, cerebrospinal fluid. Let's just get on now with a few other basic anatomical structures of the brain that I went through yesterday without a board. So I'm going to layer over this one. I'm just looking at my notes about what I covered yesterday. And let's just draw another quick outline of the brain. This is anterior, posterior. This is the cerebellum hanging down here, brain stem. Really quickly, just to reiterate, primary motor cortex, primary somatosensory cortex, and then those association areas. For example, the premotor cortex, sorry, I wanted red there, tends to run this way. In your book, they start coloring it a lighter red. And then the somatosensory association cortex is the area, the space is this way. I'm not going to go through all the nitty-gritty functions. That's all in the lecture that was recorded yesterday. The primary visual cortex is back here in this area. That would be the primary visual cortex. That's where we interpret impulses coming from the eyes, and we sort out or interpret what we're actually seeing. Primary auditory cortex in the temporal lobes in this area. That's where we sort out what we're hearing. the conversion of light wavelengths to neural impulses to action potentials actually occurs in the eyes. The conversion of sound waves in the atmosphere to action potentials occurs deep in the hearing mechanism of the ear. But the interpretation of what we hear occurs in the brain, and what we see in the brain back here. Broca's area, only on the left side, is this area of neural tissue right about here. That is predominantly focused on speech articulation. There's another area back here in the left hemisphere as well called Wernicke's area that also deals with speech. Just putting a WA for Wernicke's area. And this is speech articulation, being able to say what you want to say. Wernicke's area back here on the same side of the brain, but behind in the sort of the rear of, this is that transverse fissure. Broca's area occurs sort of above it and toward the front. Wernicke's area sort of below it and toward the back. Wernicke's area is more speech comprehension. That's understanding what somebody is saying to you, not exactly what you want to say, but they're both speech areas found in the left hemisphere. So that gives you an idea of those basic cortexes that we went over. The olfactory cortex is also found in the temporal lobe. I'm going to move on now to spinal cord anatomy. So if I get tired of talking about spinal cord anatomy, which is I think is section 1210 in your book, I'll come back and do some more repeat from yesterday just to give you more anatomical ideas of where these things exist. So now I'm just going to write, this is new. Spinal cord anatomy. I'm just going to list a lot of terms and then draw a picture of how this cross-section of the spinal cord before we leave today. So some fun facts about the spinal cord. It emanates inferior to the brainstem. So after the medulla oblongata, the spinal cord begins, if we're going from superior to inferior direction, and it extends down to about L1, it's a comma, L2 area of the vertebral column. So just if you could put your hand sort of on the top of your lower back, that's right about where the spinal cord ends. It's about 17 to 18 inches long. Its function, if you were just to describe to anybody on the street, what does the spinal cord do? What's its main function? It's a two-way street. That means a transmission pathway to and from the brain. It's also our major reflex center. I may or may not have time to go into, meaning early next week, go into how a reflex pathway occurs. What this means, a reflex, you have an idea of what that word means, I get that, but in neurological terms, what a reflex is if we have a sensory receptor somewhere out, let's just say in our appendages, and that sends an impulse, you know, an afferent impulse toward the central nervous system. So I'm just going to put SR for sensory receptor. Let's just say you're sitting in a chair and I'm tapping right below your patella, your kneecap. You know you have reflexes there. Those sensory receptors in that area just below your patella, when they are touched or tapped, They send an impulse toward the central nervous system. A reflex doesn't go all the way up to the brain to be processed. It only has to go to the spinal cord. So if this is the spinal cord, this is where a reflex gets integrated or processed, and then it immediately sends a command back out. So by sort of neurological definition, a reflex is a pathway, a neural pathway from sensory receptor to central nervous system, in this case, only the spinal cord. Doesn't have, the signal doesn't have to go all the way up to the brain to be processed. It only needs to go to the spinal cord, and then a command is immediately sent out, in this case, to skeletal muscles that makes your lower leg jerk up. So when somebody, a doctor, taps your, what that whole business is checking a reflex, is checking this pathway to see that it's working correctly. And then the skeletal muscles in your quadriceps contract, and that makes your lower leg pop up. So that just briefly explains what a reflex is. It's a shortened pathway that doesn't need to ride all the way up to the brain for processing. That's why we don't think about it either. Okay, back to the spinal cord proper itself. Spinal dura mater, spinal cord dura mater, and the dura mater is the outer thick covering, protective covering of the central nervous system. So within your reading, if you haven't encountered this yet, I'm going to explain really quickly. There's 3 meninges, M-E-N-I-N-G-E-S, 3 meninges or coverings around the central nervous system tissue. So the brain and the spinal cord. Those are called pia, it's pronounced matter, but it looks like mater. Pia mater, arachnoid matter. That gets its name because under microscopy, it looks like spider webs. Actually, what it looks like is teased cotton, like a cotton ball, the tissue. And number 3 is the dura mater. And these coverings, I should have capitalized that. They're 2 words, dura mater. These coverings, this one is the first covering that covers the neural tissue, sits right on it. It's very thin and delicate and clear. If we were to be in person doing a lab and we were dissecting a brain, or we dissect sheep brain sometimes in A&P class, you wouldn't even really see this. It's like super thin, clear plastic wrap over the brain tissue. And unless you had a probe or a scalpel and you gently picked it off, it's like a clear skin. The arachnoid matter is a bit thicker volumetrically, but it looks like a teased cotton ball. Somebody thought it looked like a spider web. That's where they gave it its name. That's the second layer outside of the pia mater, moving away from the neural tissue. And finally, the dura mater is coarse connective tissue. It really protects the brain and the spinal cord. And this is the stuff around the brain area that's attached to the inside of the cranial plates, the skull. So that gives you an idea of what those 3 layers of meninges or tissues are over the top of central nervous system tissue. Now around the spinal cord itself, the spinal dura mater, that coarse connective tissue outer covering, it's protective, is not connected to the vertebrae like it is up in the skull. So around the brain, this dura mater is connected to those cranial plates inside, but around the spinal cord, it's not connected to the vertebrae. That's what creates what we call the epidural space. If you've ever had general or almost general anesthesia, you've heard this before, an epidural injection or anesthesia. That refers to injecting anesthesia into this space that's between the dura mater and the inside of the bones of the vertebrae themselves, and it runs all the way up the vertebral column. The space in life is filled with lipid tissue. So there's fat tissue in there and mostly veins. And that's where you get anesthesia. Should you have something like a pelvic block or something like that. All right, some other terms, anatomical terms relating to the spinal cord. That's conus, C-O-N-U-S. Medullaris, that's 2 L's. The conus medullaris just refers to the end, the inferior end of the spinal cord where it comes to a point. And that happens right around L1 or L2 vertebrae. So it's just the pointy end that forms a cone of the spinal cord. This anatomical term, filum terminal, or terminal. Sometimes I've seen it spelled with an E at the end, and that's pronounced terminal. Other times without the E, that's just terminal. This is just fibrous connective tissue that connects the spinal cord at the very end to the coccyx bone. Our tailbone, the coccyx. So there's fibrous tissue emanating off of the conus medullaris that helps anchor, if this is the tailbone, which is 3 fused bones like this, the spinal cord gets anchored to that on this. It's sort of long fibrous connective tissue. These denticulate filaments just anatomical terms. We're just building a glossary, a dictionary, that's all. Are sort of these tooth-like, which is where it gets denticulate name. That's a J, projections. Tooth-like projections of the pia mater, that thin, clear, delicate connective tissue that's actually covering neural tissue itself, resting on neural tissue. And it connects the spinal cord to the dura mater. All the way down the length of the spinal cord. So all the way down from superior to inferior end. Cervical, there are things called cervical and lumbar enlargements. These are enlargements of actual neural tissue. So they're bulky areas of the spinal cord. Right directly anatomically in the areas of innervation of our appendages. So at the areas what that means is in the shoulder region, we get this bulky area of spinal cord because there's a lot of nerve roots emanating from that area that need to serve our shoulders and arms. And then there's another lumbar enlargement down at the inferior end of the spinal cord, where there are nerve roots emanating to serve our lower legs. Almost finished with this glossary. Cauda equina. Equine means horse. This is just a whole collection of nerve roots at the very base of the spinal cord. Emanating from the base, inferior end, and it sort of looks like a horse tail, which is where it got the name Cauda equina. Cauda means tail, equina means horse. Okay, now I'm going to just go to, there's 31 pairs. You don't have to memorize all of them, that's the good news. But I want you to know there are 31 pairs of spinal nerves that attach to the spinal cord at roots, what we call roots. I'm going to draw a cross-section of the spinal cord in a moment. And they're just named based on their location. For example, there's a set of first thoracic nerves, second thoracic nerves, third thoracic nerves, yada, 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 all the way down. Each nerve exits the superior end of the vertebrae. So if we were to do a dissection of the spinal cord, say you're looking at someone's backside, their back, and this is the spinal cord. If we were to dissect this or enlarge it or magnify it, each nerve exits the superior part of the vertebrae, the top part. I haven't drawn a vertebrae associated with it. So that the outgoing nerves, or the nerves serving this area of the spinal cord sort of come out of the top of that corresponding. I'm just going to draw a vertebrae here in yellow. There's the associated bone, that vertebrae in that area, and there's its spinous process, a couple transverse processes. The nerves come out or emanate from the superior part, the top of that vertebrae area. All right, let's go on to a pictorial view. So this is roughly, I'm going to try to draw a large spinal cord cross section. Just give me a moment here. This is roughly what it looks like in cross section. I'm going to use black there's gray matter and white matter. I'm going to use black to draw the pattern of gray matter in cross section. And it's important because different areas of gray matter have different functions. This is not my best artwork. But it'll have to do. So everything in here you know what gray matter and white matter is now. Gray matter, unmyelinated neuron cell bodies. White matter is myelinated neural tracts. And you know their function. So let me just label this. I'm going to label and give functions. This is all just white matter. We call these pointy areas sticking up like this the dorsal horns. because they're horn-shaped. There's one on either side. They're horn-shaped, and they emanate. Remember, this is a cross-section. So this would be the posterior side of the person, the back side, and this would be anterior. This invagination here is called the median ventral fissure. Remember, fissure is just deep groove. We also have ventral horns on either side of gray matter, that is. So there's a ventral horn. Right here I drew this circle. I'm going to color it in with blue. That is called the central canal, and cerebrospinal fluid circulates down through there. So I'm in parentheses, I'll write CSF. All right, now off to the left of this, which is why I left the left side of this picture mostly blank. I'm going to attempt to draw how the roots, the nerve roots, sort of emanate or from. So this whole thing right here we call one of those 31 spinal nerves. So I'm just going to label this spinal nerve. And there's a dorsal root and a ventral root. And I'm not even going to label this, but there's also, this thing is called the dorsal ganglion, this bulge that I've drawn. But I'm going to leave it at dorsal and ventral roots. Now I need to give you some information. I'm going to come back to this, but I'm just going to layer over some important information and then come back and help label that. So just going to layer on top of that functional statements. Now, all neurons with their cell bodies in gray matter are multipolar. You know what that means now. That's a structural classification of those nerves. Within that gray matter, the dorsal horns are all interneurons. That's a functional classification. And in the ventral horns, we have somatic motor neurons and mostly somatic motor neurons and some interneurons. Finally, in the lateral horns, which I'm not sure I labeled, I will when I go back. In the lateral horns, we have a collection of cell bodies of autonomic cell bodies of autonomic neurons, particularly sympathetic neurons. Now I'm going to just go back to this previous page, and we're just going to label this stuff. So we know the dorsal horns are all interneurons. These are functional classifications. They process information. The ventral horns down here in this area on both sides are somatic motor neurons. AKA skeletal muscle command neurons. The lateral horns, which are out here, I forgot to label those, so I'll do that now. These contain cell bodies of neurons, of autonomic neurons. I'm just writing autonomic neuron bodies, particularly sympathetic, the fight or flight arm of the autonomic nervous system. I'm just checking my notes to make sure that I have, I think so. I've labeled everything on here that you need to know. The ventral root. So I do want to label this. Functionally, this ventral root carries both somatic and autonomic neurons in it. So that's the basic gist of how this thing works, and anatomically and location-wise, where the specific functional classifications of neurons are. So the basics are, first, know gray matter from white matter. You know what gray matter is. It's the processing area of neuron cell bodies, unmyelinated neuron cell bodies. And then I want you to know the basic anatomy of this, and particularly what type of functional classifications are in each horn, as well as what resides in the central canal. If I layer anything on, I'll do that on Monday. I don't think I will. If I think of anything that I've forgotten on this basic diagram, this is the one that I usually draw when we are in person on the board, for you to get the basics of understanding of how this tissue is organized and slightly functionally what types of neurons reside in different areas of the spinal cord. So I'm going to call it there, guys. We're going to leave off with a short one. Let me cut off my recording, and then you can feel free to ask me any questions.